All right, so what I want to do today is to talk to you some more about proteins. Some of this is going to be a little bit of review. We've already talked about some of this stuff. Um, and then I want to do a little bit more demo of Chimera, specifically as it relates to visualizing protein structures. So this part should be largely review, at least this first part. Um, so I'm sure you all remember that proteins are built from smaller building blocks, which are the amino acids. There are about 21 different amino acids that are commonly found in living organisms. Here's the basic structure of the amino acid. We've talked about a little of this before, but maybe not quite this much detail. So every amino acid has a central carbon atom, which is known as the alpha carbon. Attached to that central carbon is typically just a hydrogen on one side, and then an amino group on one of the other four bonds on that alpha carbon. The amino group is NH2. And then on another one of the bonds to that alpha carbon is a carboxylic acid group. So that is carbon with a double bonded oxygen and an OH group, a hydroxyl group. And then the fourth bond on that alpha carbon, because remember, carbon always forms four bonds. The fourth bond connects to something called the side chain, represented here by the letter R. So the side chain is the part that varies from one amino acid to another. And I'll walk you through, I'll show you pictures of all of the common amino acids. I don't expect you to memorize them all, but just so you're aware of how different the different amino acids are based on that side chain. So amino acids actually get their name from the fact that they contain both an amino group on one side and an acid group or carboxylic acid group on the other side. The 21 amino acids are subdivided into several different groups based on their chemistry, based on the chemistry of the side chain. And one of the big distinctions about the side chains of the amino acids is whether the side chain is polar or non-polar. So a non-polar side chain is a group of atoms attached to the amino acid that it has no net charge one way or the other. It's not slightly more positive, it's not slightly more negative, it's completely electrically neutral. One consequence of that is that any of the amino acids that have these non-polar side chains tend to be repelled by water. So they actually try to migrate towards the center of the protein to get away from any water surrounding the protein, whether it's cytoplasm or extracellular fluid. So these are the amino acids that have non-polar side chains. And again, I don't expect you to memorize all of this, but if we look at the simplest one over here on the far left, this is glycine. So that carbon that I'm circling right now, that's the alpha carbon at the center of the amino acid. It has an H next to it, indicating the hydrogen that's attached to one of the bonds on that alpha carbon. On the left is the H2N or NH2, that's the amino group. On the right is the COOH, the carboxylic acid group. And then the side chain for glycine is literally just another hydrogen atom. So it's by far the simplest of all of the amino acids. The next one here, alanine, again, there's that alpha carbon, and the red represents the side chain. In the case of alanine, the side chain is simply a methyl group, CH3. And then you see that we get some others like valine and leucine and isoleucine that have slightly more complicated side chains, but they're basically just branched chains of carbon atoms. They're completely non-polar. There's no slight positive or negative charge or anything like that. So these tend to be repelled by water. Some of them get a little more complicated, like phenylalanine. It's the same structure as alanine with that CH3 or methyl group, but now we've added an entire benzene ring to that methyl group. So the phenyl is a prefix that often refers to these benzene rings. So it's simply phenyl added to alanine. Tryptophan, you probably heard a lot about this one. This is the one where there's that myth about the reason you get sleepy on Thanksgiving is because of all of the tryptophan in the turkey. Well, guess what? Every living organism contains a lot of tryptophan Every animal contains a lot of tryptophan. There's no more tryptophan in turkey than there is in ham or pork or whatever, or beef. And you'd have to eat so much tryptophan. You'd have to eat like, 40 pounds of turkey or something like that for it to actually make you sleepy. So the real reason you get sleepy on Thanksgiving is because we tend to eat way too much and sit around watching football and not getting any activity. But this is the structure of tryptophan. You see it's got a five-sided ring and then a benzene ring attached to it. One of the more unusual amino acids is proline. This one actually doesn't have the classic structure of an amino acid. The alpha carbon is actually part of this ring, this five-sided ring down here. And then it doesn't have a true amino group. Instead, it just has this HN over here, and then the carboxylic acid part up at the top of the ring. It still participates in the formation of proteins. This HN can still form what's called a peptide bond. The OH on that carboxylic acid can also form a peptide bond. So proline can be incorporated into an amino acid chain, just like any other amino acid, but it actually creates a kink or a bend in the amino acid chain because it has this kind of unusual structure to it. So these are the nonpolar amino acids where the side chain has no electrical polarity. And I think I talked briefly about this when I talked about color schemes, that there's a color scheme for actually coloring the different amino acids, and it has a lot to do with the charge or the polarity of the side chain. All of the nonpolar amino acids use dull colors to represent the fact that they have no polarity. At the opposite extreme, we have some polar side chains that actually have an entire +1 or -1 charge. So they actually form ions and have a full charge, either -1 or +1. And these are represented by bright colors to represent the fact that they're very polar. Anything that has a net negative charge, like the two on the left, is colored bright red. And the reason for that is because the negative charge in these two amino acids is coming from oxygen. And since oxygen is colored red in the CPK color scheme, these amino acids are colored red in the amino acid color scheme. So these two are glutamic acid and aspartic acid. So they have the standard amino acid structure. There's the alpha carbon in the center with a hydrogen attached to it, the amino group on the left, and COOH, the carboxylic acid, on the right. But these then have another carboxylic acid in their side chain. So these are almost like a double acid. And the hydrogen atom tends to split away, leaving that oxygen with an exposed negative charge on it. So that's glutamic acid and then aspartic acid. And we also have some amino acids that will end up as positively charged ions or cations. And the positive charge is always coming from nitrogen. And since nitrogen is always blue in the CPK color scheme, these amino acids are always colored blue in this amino acid coloring scheme. So these are lysine and arginine, and you can see that they have another amino group, NH2, but that amino group tends to attract an additional hydrogen, giving it a net positive charge. There are also a bunch of amino acids where the side chain has an electrical polarity, meaning it's a little bit more positive on one side, a little bit more negative on the other side. So we call them polar, but it's not a full-blown charge. It's not a full +1 or -1, but there is still a slight polarity, electrical polarity, to the side chain. These are also, they're hydrophilic, they're attracted to water, so they tend to be attracted to the outside of the protein. And we don't represent them with vivid red or vivid blue, but we still use warm or cool colors. The warm colors are used for the ones that are maybe slightly negative in polarity, because that negative polarity is usually coming from oxygen. So we use warm colors that are similar to red, like oranges and pinks and things like that. And then we use cool colors for the ones that have maybe a slight positive polarity. Again, not a full positive charge, but still maybe slightly more positive than the surroundings. And that positive polarity is usually coming from nitrogen. So again, we use cool colors similar to the blue of the nitrogen. So serine and threonine and tyrosine, they all have OH groups in them. And those OH groups tend to make them slightly more negative. So they have a slight negative polarity. The ones down at the bottom, histidine, glutamine, and asparagine, all have nitrogen in their side chains, which tend to make them slightly positive polarity. So those are to be represented with cool colors. And then finally, there are two amino acids that actually have sulfur, a sulfur atom in their side chain, cysteine and methionine. And because sulfur is always yellow in the CPK color scheme, these amino acids are colored yellow if we were to view a protein where it was colored by its individual amino acids. So I will be quizzing you about proteins at some point in the near future, but I'm not going to expect you to memorize all of these structures. I certainly haven't. I mean, I know a handful of them, but I don't know them all by any means. There are shorthand codes, abbreviations for all of these amino acids. And so sometimes when you see the amino acid sequence for a protein, you'll actually see it written out as a series of abbreviations rather than writing out the full names of every amino acid, which would be very cumbersome. So every amino acid gets a 3-letter code. And then these I actually have memorized, but I don't expect you to memorize these either. Just, you may eventually, if you work with this stuff enough, you may get to the point where you start to recognize some of these three-letter codes. Like alanine's pretty easy because it's literally the first 3 letters, and cysteine and aspartic acid and glutamic acid. A lot of these, they just take the first 3 letters, so they're relatively simple. In addition to the 3-letter code, every amino acid also gets a 1-letter code. And this I absolutely have never tried to memorize. I've never bothered. But I do know that occasionally you'll see the amino acid sequence of a protein listed as just a series of single letters, where each one is assigned to a specific amino acid. Some of them are easy, like M for methionine, A for alanine, C for cysteine, but then you get into a lot of weird ones like W for tryptophan. I guess, you've only got 26 letters, so you got 21 amino acids, so it's basically 1 letter per amino acid. Right, so we've talked a little bit about how amino acids actually join together, but just let's review that again quickly. So we've got your amino acid with the amino group on one side, the alpha carbon in the center, carboxylic acid on the other side, and then that side chain, that variable side chain kind of sticking off to the side. Amino acids form proteins at the ribosome, right? So this is the process of translation where you have a messenger RNA template, and then you have tRNA, transfer RNA molecules, come in and bind to that mRNA template, and they deliver individual amino acids to the ribosome. And those tRNA molecules end up holding the amino acids very close to one another in close proximity, so that the carboxylic acid of the very first amino acid is held right next to the amino group of the second amino acid in the chain. And by holding these in close proximity, a chemical reaction occurs. The OH from the carboxylic acid breaks off. One of the hydrogens from the amino group breaks off. Those split away to become water. So OH plus H is H2O. And the carbon from the carboxylic acid ends up binding directly to the nitrogen of the amino group. And that forms what we call a peptide bond. And the new molecule that's created is called a dipeptide because it consists of 2 amino acids joined together. And then another amino acid would be delivered to the ribosome by another tRNA molecule. That would be held so that its amino group is adjacent to the carboxylic acid group of the one before it. The same reaction would occur. The OH from this amino acid would break off along with the hydrogen from from the amino group of the next amino acid, and you get another peptide bond formed. So now you'd have a tripeptide, and this continues to happen always adding new amino acids to the carboxylic acid end of the amino acid chain. And eventually you get a long line of these, a long chain of these amino acids. So down here, the alternating colors, the green, the yellow, the green, the yellow, represent the amino acids after they've been joined together in this long polypeptide chain. There's a term that you may see used frequently if you deal with, having to illustrate proteins. They may refer to something as a residue. A residue simply means each amino acid in the chain after it's bonded to its neighbors. Because technically, once it's incorporated into the peptide chain, it's not really an amino acid anymore. It's sort of a remnant or residue of an amino acid. So each amino acid, once it's part of the polypeptide chain, is referred to as a residue. Technically, the definition of a protein is it's any polypeptide chain with more than 50 residues. But in reality, that definition doesn't really hold up because I can think of at least one protein that has fewer than 50 residues, and we still call it a protein. So really, a protein is just any long chain of amino acids, any polypeptide chain. Remember that the very first amino acid in the chain had its amino group kind of at the beginning of the chain, and then all additional amino acids are always added to the carboxylic acid end. That means that every amino acid chain has a direction to it. It has a start and it has an end. The start being where the very first amino acid was laid down at the ribosome, and the end being where the very last amino acid was laid down at the ribosome. These are referred to as the N terminus and the C terminus. The N terminus because that's the amino group end of the very first amino acid, and the C terminus because that's the carboxylic acid or carbon end of the very last amino acid. So be aware that every protein chain is going to have an N terminus at its beginning and a C terminus at its end, representing the order in which the amino acids were laid down at the ribosome. Right, so we've talked a little bit before also about protein structure, although I'm adding an extra one in here, an extra level of protein structure. So you may remember we talked about the fact that every protein has a primary structure, which just refers to the linear sequence of amino acids basically forming a straight line of amino acid residues. That straight line of amino acids then begins to spontaneously fold to form various kinds of helices or coils and zigzags back and forth on itself to form sheets like beta sheets and various other kinds of loops and other structures. These are all considered part of what we call the secondary structure of the protein, where that amino acid chain then begins to coil or fold. So things like alpha helices and beta sheets. There's another level of structure that we didn't talk about before that's kind of above secondary, but not quite tertiary yet. So these are called super secondary structures. You can think of it as being like 2.5 degrees instead of, it's not 2, it's not 3, it's kind of in between. What happens in a super secondary structure, and I'll show you some examples of these in a minute, is the secondary structures then fold to form structures that are a little bit more complex than just the basic secondary structure. But the protein hasn't yet completely folded into a compact blob, so it's not quite at the level of tertiary structure either. So it's kind of in between secondary and tertiary. So then all of these secondary structures and supersecondary structures then fold to form a compact blob of some kind, and we would call this the tertiary structure of the protein. And for a lot of proteins, that's it. That once they reach their tertiary structure, they're done. They are now functional proteins. So myoglobin is a good example of that. Myoglobin is just a single amino acid chain that forms a primary structure, secondary, supersecondary, and then all of that folds into a compact blob to form a functional molecule of myoglobin. But then there are other proteins that consist of multiple amino acid chains that were produced independently at the ribosome. And these different amino acid chains then have to come together to form an even more complex structure in order for the protein to be functional. And this is what we call quaternary structure. So that's where you have a group of tertiary structures each one representing a different amino acid chain that then have to join together to form a single functioning protein. So a classic example of this is hemoglobin. Hemoglobin is almost like four myoglobin chains that join together to form a single functioning hemoglobin molecule. So here's a visual representation of the primary structure of a protein, simply this linear chain of amino acids. And you can see that there's the backbone, which is the alternating nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon of each of the amino acid residues. And then all of these little balls here represent the side chains of the individual amino acids hanging off of the side of this amino acid chain. That amino acid chain then either coils or folds to form these secondary structures, which we've talked about some of these before. So in the alpha helix, the amino acid chain forms a spiral, and the spiral is stabilized by the presence of hydrogen bonds, which are weak attractions between the negatively charged oxygens and the positively charged hydrogens on different amino acids in the chain. Another type of secondary structure is the beta sheet. We've talked a little bit about beta sheets, but we didn't talk about the fact that there are two broad categories of beta sheets. The most common type of beta sheet is the bottom one here, called an anti-parallel beta sheet. Anti-parallel meaning not parallel. So what's happening here is we've got the amino acid chain going, in the direction of this arrow. It then makes a tight 180-degree turn, turns back on itself, and has another straight segment, and then makes another 180-degree hairpin turn and comes back the other way, and then again another 180-degree hairpin turn, comes back the other way. So in this particular example, we've got four strands of the amino acid chain, all side by side, and they're all pointing different directions. The arrowheads are pointing in the direction from the N terminus towards the C terminus. So that's what the significance of the arrowheads is. And these segments of the amino acid chain are then held together by hydrogen bonds represented by the little dots. And this then ends up forming a broad flat sheet of the amino acid chain. The other kind of beta sheet is what's called a parallel beta sheet, and these are a little bit less common. In a parallel beta sheet, you've got the amino acid chain going one direction, and then it kind of loops around and zigzags around for a while, and then comes back and forms another segment that's running in the same direction as the original segment. And then it loops and squiggles around some more, comes back, forms another strand facing in the same direction. So you can see here we've got four strands, but all four of these strands are facing in the same direction. They're all pointing the same way, meaning they're all pointing in the same direction from N terminus towards C terminus. And just like with the anti-parallel beta sheet, those different strands are stabilized together by hydrogen bonds. Those tight hairpin turns that you particularly see in the anti-parallel beta sheet, those are called beta turns because it's where one part of the beta sheet turns to become the next part of the beta sheet. So that's also considered a component of secondary structure. And then there are various other things that don't really have any particular structure to them or no repetition, various kinds of loops and bulges and just random strands of the chain that don't have any particular structure to them. So then we get to these things that we haven't talked about before, these super secondary structures, or sometimes they're referred to as motifs. So it's a little bit more complicated than secondary structure, but still not as complicated as tertiary structure. So these are just a couple. There are a bunch of others that I didn't bother to show here, but there's something called a beta alpha beta unit. So it's a component of a beta strand or beta sheet that's pointing in the direction from N terminus to C terminus. It makes a tight turn. Then there's an alpha helix, and then there's another strand that's part of another beta sheet. So it goes beta sheet, alpha helix, beta sheet. And this is actually a fairly common motif that's found in a lot of proteins. And the purple lines here just kind of represent the zigzagging of the beta sheet, alpha helix, beta sheet. There's another one called the Greek key, which is a type of anti-parallel beta sheet, but where the individual strands of the beta sheet are joined together in kind of an unusual pattern. I've always thought it looked like a paper clip. How you take basically a straight piece of wire and you fold it, and then you fold it back on itself to create the folding of the wire in a paper clip. And you can see the purple lines just kind of represent how that particular amino acid chain is folding. It's called a Greek key because it's actually a very common pattern seen in a lot of Greek pottery. Finally, there's something here called a beta barrel, which is a beta sheet, but the whole beta sheet has then wrapped around 360 degrees to form a complete cylinder, right? So just imagine a flat sheet of paper, which would be your beta sheet, and then you just fold that sheet of paper around to make it a tube, a cylinder. So then from our secondary and supersecondary structures, we jump up to the level of tertiary structure. So this is where all of the secondary and supersecondary structures join together to form a compact blob. We've seen this thing before. This is John Kendrew's low resolution model of myoglobin, where each of these white sausages is actually an alpha helix. And then those alpha helices are all tied in a knot here, all folded up into this compact blob, which is the tertiary structure of myoglobin. So this would be a fully functional molecule of myoglobin at this point, once it's folded into this tertiary structure. And then we've seen this before, the classic example of quaternary structure, where this is Max Peritz's model of hemoglobin. So in a quaternary structure, you have multiple amino acid chains, each of which is forming a tertiary structure, but then those tertiary structures all have to come together to form a more complex molecule, which would be what we'd call a quaternary structure. So in order to have a functioning molecule of hemoglobin, you actually have to have 4 separate chains joining together to form a single hemoglobin molecule. All right, so there are different ways of representing proteins. And again, a lot of this is going to be largely review from before when we talked about, representing all kinds of molecules. So one of the simplest ways of representing a protein would be with just a simple wireframe model, or what we call a stick model or ball and stick model. So this would be kind of like the high resolution model of myoglobin that John Kendrew created, where he had all of these little individual sticks representing every single bond between the atoms in the protein chain. And you can see that it really just looks like kind of a random jumble until you put the piece of white string in there and you realize that that's actually the folding of the amino acid chain. And on the right, we have the painting by Irving Geis of that model. You can represent proteins as space-filling models or CPK models, where every atom is represented by a sphere that is the size of its van der Waals radius. And this starts to give you a sense for the overall 3-dimensional structure of the protein. So those are representations that you could really use for any kind of molecule, and you could certainly use them for proteins too if you wanted to. But then there are other special representations that are specific to proteins. So remembering that every protein is made of a polypeptide chain, a chain of amino acids, oftentimes it's very important to understand how does that linear chain of amino acids end up folding to form the final structure of the protein. So people who study proteins develop this thing called the backbone model that only looks at the bonds between the nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, representing the individual amino acid residues. You look only at that strand of those 3 atoms, which represents the amino acid chain, and you simply ignore everything else. You hide the double bonded oxygens, you hide the hydrogens, and most importantly, you hide the side chains, the R groups. So in this picture that I've shown you before, the backbone model of the protein would simply look at that gray bar and ignore all of those side chains hanging off the sides of the amino acid chain. So a backbone model would look something like this. So this is a backbone model of the protein called lysozyme. And what we're looking, each little straight segment represents nitrogen, carbon, carbon of a single amino acid. So nitrogen, carbon, carbon, Nitrogen, carbon, carbon, nitrogen, carbon, carbon for each little segment. And all of the double bonded oxygens, the hydrogens, and the side chains of all the amino acids are hidden. But it allows you to easily see how the amino acid chain folds to form this compact blob. A slight variation on that, another version of a backbone model is the ribbon model that emphasizes the secondary structures present in the protein. So it's still showing us the folding of the amino acid chain as it passes through the center of this compact blob of a protein, but it identifies which parts of that amino acid chain are actually forming alpha helices, which part are forming beta sheets, and which parts are just forming sort of random loops and coils. So you see this protein on the left has 3 different alpha helices, big one at the bottom, big one on the left, and kind of a small one hiding in the back there. And it has a beta sheet. This would be an anti-parallel beta sheet where the arrowheads are all pointing different directions. On the right, here's another protein that has a couple of alpha helices. It has this very strange beta sheet where the whole beta sheet is kind of twisted. It's almost like if you took a piece of paper and kind of twisted it. So here's a 3D model of a protein represented as a ribbon model. This is that same lysozyme protein again, and you can see the coils for the alpha helices, and there's a few strands in the background which are part of a beta sheet. Here's another ribbon representation. This time we're looking at potassium channel. This one doesn't have any beta sheets in it. It's all alpha helices, plus then some random loops connecting the alpha helices together. Another type of model, which really you could use it for any kind of molecule, but it's most commonly used for visualizing proteins, is this solvent accessible surface model that we talked a little bit about before. And what this tries to do is it tries to show you where the deep indentations are in the surface molecule, the surface of the molecule. It just kind of glosses over any small little pockets or small indentations because they're not big enough to really allow anything to get inside. But what this model is going to show is, are there any big cavities, any big pockets on the surface of the molecule that could serve as binding sites for some smaller molecule to bind to the protein. So here we see a comparison between a typical space filling or CPK model of lysozyme on the left, and then the exact same molecule represented as a solvent accessible surface. And so it smooths out much of the surface, but it still allows you to see where some of the larger pockets and indentations are, which may be important binding sites for this protein to do its job. And I think I've shown you this before. This is the surface model of that same potassium channel that I showed you just a minute ago. And this one does have a binding site somewhere up here because it actually binds to a ligand, which then opens the potassium channel. If I could rotate this around, if it were a 3D model, you'd be able to tell that there's actually a pore, a hole, or a channel that runs the length of the protein, which is the channel that allows potassium ions to actually pass through this particular channel. Here's another variation on the surface diagram. These are some of the paintings by David Goodsell, who I've talked about in here a little bit before. I showed you his book way back on the first day of class. And he's developed this very, very distinctive style. I mean, it's so distinctive that there's actually at least one molecular visualization app that allows you to view things in the Goodsell style. It actually has a choice called, viewing things in a David Goodsell style. So it's kind of like a solvent accessible surface where things have been smoothed out a little bit. You can kind of see every individual atom, but they're not colored like in the CPK color scheme. So it produces a very, very clear image where you can clearly see, that these things are made up of individual atoms, but you're not losing the forest for the trees. You can still get a sense for the overall form of the molecule. You may not remember it, but way back on day one, I also showed you a book It was a molecular cell biology book. And it was illustrated by this guy, Graham Johnson, who's a graduate of the Hopkins program. And when he was tasked with illustrating this molecular cell biology book or cell bio book, he kind of adopted the David Goodsell style and modified it a little bit. So these are things that are all rendered in Cinema 4D using a kind of a Goodsell style of rendering for these different proteins. So there are also these different color schemes for depicting proteins. And again, this will be largely review, or at least partly review. So if you represent a protein as a stick model or ball and stick or a space filling model like this one, then you can use the CPK color scheme where you get gray carbons, blue nitrogens, red oxygens, and white hydrogens. But there's gray carbon, blue nitrogen, red oxygen, and white hydrogen. Sure. There's also this amino acid color scheme where every amino acid in the protein gets a different color, and it's all based on the polarity or lack of polarity of the side chain. So we talked about this briefly before when I was showing you the different amino acids. If an amino acid has a non-polar side chain, it's represented by a dull color, like this dark gray, or the unfortunately called flesh color, or tan, or light gray. If something has a polar side chain, it's represented by a bright color. And if that polarity is a negative polarity coming from oxygen, then we color it either in solid red or another warm color like bright red or orange. If the polarity in the side chain is more of a positive polarity, it's always coming from nitrogen, so we're going to color them blue or a cool color. So lysine and arginine are the two amino acids that actually have a full positive charge because of a nitrogen in the side chain, so we color them this blue. But then things like mid-blue and cyan would come from, would also have a polar side chain where that's coming from nitrogen. And then there are the two amino acids that have sulfur in their side chain, cysteine and methionine, that would be colored yellow. And so you can actually take something like a space-filling model of a protein. This is that same lysozyme, one more time. But in this case, I applied an amino acid color scheme to it. So you're still seeing the individual atoms, but you can see how they're clustered. So there's a cluster of atoms right here that are all bright red. So I could say pretty confidently that that's either aspartic acid or glutamic acid, because those are the only amino acids that would be colored red like this because they have a full -1 negative charge. There's some orange ones in here, which would be warmer colors, but so they wouldn't have a full -1 charge, but they'd have a slight negative polarity. There's also some bright blue ones. These would be the ones that have a full positive charge because of nitrogen in their side chain. So that would be lysine and arginine would be the bright blue ones. And then anything that's light gray or dark gray, these would be things that would have non-polar side chains like glycine, leucine, isoleucine, some of these other non-polar ones. You can also take a ribbon model, and kind of depends on the software. I don't know if all of the visualization applications let you do this. These were actually created in that really ancient program that I talked about a little bit called Wetland Viewer Light. But so here we have a ribbon model. The little bands of color actually represent the individual amino acids, and they're colorized according to that same color scheme. So this dark blue one here would be either lysine or arginine. Looks like there's some yellow ones, which would probably be the sulfur-containing ones, like cysteine and methionine. If you represent a protein by its secondary structure, where we're seeing the alpha helices and beta sheets, you can choose to color the protein by secondary structure. So you can apply one color to the alpha helices, like the color red in this case, and a different color for any beta sheets that are present. So in this case, that turquoise represents a beta sheet. So it makes it very easy to distinguish the different elements of secondary structure in this particular protein. And the little gray parts are just the random loops and coils that don't really have a true secondary structure. Ignore the space filling models. Those are not part of the protein itself. Those are actually, this thing over here is GTP, guanosine triphosphate, which is always incorporated into this particular protein. The thing on the left is actually a drug molecule. It's a chemotherapy drug molecule. So I was actually creating this illustration focusing on the action of that chemotherapy drug in this particular protein. But they're not part of the protein itself, which is why I rendered them in a completely different style. If you have a protein that has quaternary structure, remember, quaternary structure means a protein that has multiple different amino acid chains that have to come together to form a functional molecule. You can choose to color the molecule by chain to distinguish the different chains. So this is a model of immunoglobulin G, or IgG. This is the classic Y-shaped antibody. Oftentimes you'll see like immunology illustrations where they'll represent it as just a simple little Y shape. But this is the actual three-dimensional shape of an IgG antibody. And it consists of 4 separate amino acid chains that have been colored separately in this illustration. So there's a red chain, which starts way down at the bottom and comes up and then goes out into this right-hand arm. And then there's an orange one that also starts at the bottom and goes off into the left-hand arm. Those are called the heavy chains of the antibody. And then the blue one and the turquoise one, those are referred to as light chains. So every antibody has 2 heavy chains and 2 light chains. And in this case, they're distinguished from one another by their colors. All right, so some new stuff is just talking a little bit about the different kinds of proteins that are out there that exist in nature, and a little bit about their function. So proteins are first subdivided into 2 very broad categories. There are the fibrous proteins, which, as the name suggests, tend to exist in long fibers. They're often structural proteins that give things strength. And then there are the globular or compact proteins that tend to exist in more of a blob shape. So the fibrous proteins tend to have long chains, and sometimes these chains are literally just alpha helices, just really, really long continuous alpha helices. And keratin is a good example of that. So keratin, the protein that forms hair, so human hair and then any kind of animal fur, but it's the protein that forms feathers in birds. It's the protein that forms antlers in deer, rhinoceros horn, fingernails, toenails. So a lot of things in the skin, hair, nails, feathers, that sort of thing. And like I say, it's basically just long alpha helices. There are some other structural proteins, some other fibrous proteins like collagen, which is found in the skin, which is not just a simple alpha helix, it's more of a braided. I think it's like 3 alpha helices that are intertwined and braided together. Then the globular proteins, or sometimes just referred to as compact proteins, these are further subdivided into several different types. There's lots of globular proteins that are called membrane-bound proteins. So these are all of your proteins that are embedded in the cell membrane. So most receptors are going to be embedded in the cell membrane. so that they're actually accessible to, whatever's trying to bind to that receptor. Some ligand is coming in from, from the extracellular fluid or from the bloodstream or wherever. And so this protein is actually embedded in the cell membrane, so it's accessible to that ligand to bind. Channel proteins, so anything that allows something to pass through the cell membrane. These are things that are also going to be membrane proteins that are embedded in the cell membrane. Enzymes, I think all enzymes are going to fall under this globular or compact protein category. So these are all of these proteins that have some kind of binding site on them where they're going to interact with some other chemicals and either take a larger chemical and split it into two smaller pieces, or take two small molecules and bond them together into a larger molecule. The transport proteins, things like hemoglobin and myoglobin, that transport and store oxygen. But there's other kinds of transport proteins, cytochrome C, a lot of the proteins that are embedded in the membrane of the mitochondria. There are 4 different groups of proteins embedded in the membrane of the mitochondria that are responsible for the generation of ATP. These are also considered transport proteins because they're actually transporting electrons as part of what we call the electron transport chain. And then there's all kinds of other miscellaneous compact proteins or globular proteins. So the antibodies, I showed you an example of that already. That's a type of compact protein that circulates throughout your bloodstream and in your bodily fluids to combat infections. Histones, which are a group of proteins that DNA wraps around to form chromatin. I'll show you a picture of that in just a couple minutes. So let's take a look at some pictures of some of these things. We'll start out with just one example of a fibrous protein. This is the 3D structure of keratin, and you might not be able to tell right away, but this is actually an alpha helix. So what we're looking at here is we've got a carbon atom at this corner point, another carbon atom at this corner point, and then a nitrogen. So it's going carbon, carbon, nitrogen, and then we go to another carbon, carbon, nitrogen, carbon, carbon, nitrogen. So this is simply the repetition of the amino acid chain, but it's been coiled into a spiral. So this is actually what an alpha helix looks like if you were to view it as a simple stick model. So jumping to the compact or globular proteins, here are some examples of some membrane-bound proteins. The layers of white balls represent the outer and inner layers of the cell membrane. So the outer and inner layers of phospholipids that make up the lipid bilayer of the cell membrane. So on the left is the acetylcholine receptor. It's actually one type of acetylcholine receptor because you actually have a couple different kinds. This is what's known as a nicotinic acetylcholine receptor. So somewhere out here on the external part of this receptor is where acetylcholine would bind. And then what that does is it actually opens up a channel that allows sodium ions to then pass right through the center of the protein to have a rapid influx of sodium ions into the inside of the cell to depolarize the cell membrane. On the right is an example of a potassium channel. Again, there are many, many different kinds of potassium channels, so this is just one of them. It's also embedded in the cell membrane, the 2 layers of phospholipids. The little turquoise balls in there, those actually represent potassiums that are waiting for their turn to pass through this channel, to pass from one side of the cell membrane to the other. Here's an example of an enzyme. Not very exciting looking thing. I mean, it's just a blob. But this blob has 2 very distinctive pockets on one surface, which house heme groups. So interestingly, this particular enzyme contains two heme groups, which is the same thing that holds the oxygen atom in the hemoglobin molecule, and also in myoglobin as well. And this is the active site. These are the active sites for this particular enzyme. This is an enzyme called cyclooxygenase. It's an enzyme that converts a lipid that's found in the lipid bilayer of your cell membranes. There's a lipid called arachidonic acid, which is found in the cell membrane of, I think, basically all living things. And if there's damage to any of your cells that disrupts the cell membrane, it releases these molecules of arachidonic acid into the extracellular fluid, where it binds to this enzyme, and the enzyme converts that lipid molecule into another category of molecules called a prostaglandin, which is a powerful mediator of pain and inflammation. So this is one of the primary enzymes that's responsible for pain and inflammation after any kind of injury. So those prostaglandins are going to recruit various kinds of white blood cells to the area. They're going to cause dilation of vessels and leakiness of capillaries, so you get swelling, edema, accumulation of fluid in the tissues in response to an injury. So that's why, anytime you injure anything, it swells. And those prostaglandins also then trigger free nerve endings to create pain. So that's where most pain comes from, is from prostaglandins stimulating free nerve endings after an injury. And there are several kinds of drugs that are designed specifically to block this enzyme. Aspirin, ibuprofen, acetaminophen, and a whole new category of drugs that are selective cyclooxygenase inhibitors. They're called COX-2 inhibitors. You may not be familiar with these because I think most of them are prescription, probably all prescription. There's a drug called Celebrex, which is a very powerful painkiller. And anyway, so they're all targeting this particular enzyme to prevent the production of prostaglandins. I already showed you this picture. Here's an immunoglobulin. It's just another example of a compact type of protein. This one here, it may be a little bit hard to actually see the proteins because they're all represented as ribbon models. But what we're looking at here, if you look at this bracket and you look at the label just to the left of that bracket, we're looking at histones. So all of those little spirals that you're seeing, those little corkscrews, those are all alpha helices. So there's a series of proteins in here in all different colors. In fact, there's 8 of them. And I don't know if we can see all 8 colors, but there's a yellow one, there's a green one, there's kind of a gold one, there's a turquoise one, there's a red one up there, there's a purple one over here. There should be 8 different proteins in there that are all called histones, and they form what's called a histone octamer, which just means 8 of them in a cluster. And the DNA double helix then coils itself around this octamer, this group of 8 histone proteins. And if you take 1 3/4 turns of the double helix around those histones, so we've got like 1 complete turn and then about 3/4 of another turn, that whole complex, the 8 histone proteins, and then 1 3/4 complete turns of the DNA double helix, that forms a unit known as a nucleosome. These nucleosomes are then packed when it condenses into chromosomes. A little bit later in this course, I'm going to give you another lecture specifically about nucleic acids. And there's a beautiful animation that shows how the DNA coils around histones to form a nucleosome, and then how the nucleosomes are packaged together to form chromatin, and then how the chromatin and then in turn is then coiled further to actually form a full chromosome. All right, one last point I want to make before we take a little break is that proteins are not static. And I'm going to have you go to a couple of websites to retrieve proteins, not necessarily today, but for our next couple of illustration assignments, I'm going to have you go to websites like the Protein Data Bank and download 3D models of proteins that have been solved using X-ray crystallography. And you're going to open up these proteins and you're going to get a static three-dimensional model of a protein. It's very challenging. You have to have a very sophisticated knowledge of 3D software. You have to have a very sophisticated knowledge of molecular biology to actually show how these proteins change shape over time. We're not going to actually get into that level of detail in here. I just want you to be aware of the fact that proteins don't always just have one shape, that they change a lot. And I'm going to actually show you a couple of videos that are going to point this out. What you're looking at here are the coronavirus spike proteins. And you may remember from way back when we created our own coronavirus, we were searching this electron microscopy database, and there were hundreds of these things, right? There were hundreds of different versions of these coronavirus spike proteins. And you might remember I said one thing to watch out for is that there are two different configurations. There's the closed configuration and the open configuration. And if you're just showing a coronavirus that's like floating around in somebody's droplet of nasal mucus, they're all going to be in the closed configuration, which is the picture on the right. It's only when that spike protein gets close to its receptor, which is ACE2, angiotensin converting enzyme type 2. It's only when it gets close to that receptor that it changes to the open configuration. But then after that, it gets crazy. I'm going to show you these beautifully done animations that show you just how much a protein will change shape. I don't think there's any audio. But what you're looking at is the spike protein. And here the spike protein just opened. And it's, all right, this is supposed to stop. Why are you not stopping? I was trying to pause and go back here. All right. So the spike protein was in its closed state. And as it encounters this ACE2 receptor, that's when the spike protein is going to switch to its open configuration. So it's just starting to open. There's the open configuration now. That's going to allow it to bind to ACE2, but then it's going to do something even more dramatic. The ACE2 is going to rip off the pink part of the spike protein, exposing only the yellow part. And that's the part that actually allows the virus to bind to your cells. So those are all alpha helices that are unfolding themselves. So this was created by somebody who I've mentioned in here before, although you probably don't remember the name, Gail McGill. He has a PhD in molecular biology, so he really understands how this stuff works. And he actually teaches molecular biology at Harvard, at Harvard Medical School. But he also runs this company called Digizyme. And so he has a team of animators who work closely with him to animate this kind of stuff. So he does some very sophisticated high-end molecular animations. He's also the guy who invented molecular Maya, which we don't use in here, but those of you who use Maya more may want to try to explore that. And now I got another one showing pretty much the same thing. It's just, I think this one actually has narration, maybe a little bit more explanatory. And
Zero helps me take my business anywhere I want.
oh no, thank you. Okay.
The surface of the virus is studied with spike proteins, colored teal, membrane proteins, colored yellow, and envelope proteins, colored red. Cross-section shows the viral RNA genome orange, and nucleocapsid protein, which protects the RNA, gray. Be infected with SARS-CoV-2 after inhaling airborne viral particles. On entering the airways, the virus particles encounter respiratory epithelial cells in the trachea and lungs. The SARS-CoV-2 spike protein is a membrane-bound protein at the surface of the cells known as ACE2.
So all those little purple guys, those are the ACE2s.
Shown here in purple.
Oh, sorry.
Once bound to ACE2, the spike protein is unable to open, close, and become freely, locking it in place. Another cell surface protein, transmembrane protease serine 2, shown here in orange, cuts the spike protein in specific location. These segments of the spike protein fall away, expose previously hidden parts of the spike. The spike protein then undergo a series of dramatic conformational changes. During the first stage, the spike protein inserts itself into the cell membrane. Once inserted, the spike protein folds back on itself, pulling the membranes of the cell and virus together so they can fuse. After fusion, viral RNA is deposited into the host cell, where it hijacks the cell's machinery to produce more virus particles.
All right, what I want to do for the remainder of our time today, or hopefully it won't take up the whole time, but I want to show you some more of the functionality of Chimera. And I'm going to show it to you by showing you specifically things that relate to proteins and visualization of proteins. So first of all, I'm going to start off by opening just some models of the individual amino acids. And I've got my computer set up to it launches Chimera directly if I click on the right kind of file. So this is a model of alanine, which is one of the simpler amino acids, and I'm going to try to orient this in such a way that it kind of matches the picture that I showed you before. Remember, under the actions menu, you can change different kinds of representations. You can change colors. I'm going to color this by element because I don't like the tan for carbon. And I'm going to change our depiction of the atoms from stick to ball and stick. Oh, that's interesting. Got little tiny hydrogens.
Jim, should we be doing this on our computers as well?
You don't have to.
Okay.
No. Wonder where that setting is. It's making those hydrogens so darn small. I'll have to play around with that. I don't want to take the time to do that now. Actually, the carbons everything's unusually small. That's really weird. Wonder if that's something in the settings. Yeah, this is kind of annoying, so I want to make sure to change that. That might have something to do with it. I don't know how that got changed. Okay. Well, let me see if this helps at all. Oh, that's maybe slightly better, but not much. It should be Okay, that's weird. It keeps resetting it to 0.15 for the ball scale. Let's try this. Okay, now it's huge. That's a sphere model. That's not a. All right. I think I may have gone overboard. Let's go back here. Try that again. Hopefully, third time's a charm. Let's see what happens. That's better. That's what I want. Yeah, so I must have messed around with the settings in there or something got glitchy. But all right, so I'm going to try to orient this similar to the picture that I had of the amino acids in the PowerPoint. Whoa. Um, so that carbon right in the very center, that's the alpha carbon. And it's connected to a hydrogen, which is facing away from us at this point. And on the left-hand side is NH2. Nope, sorry, strike that. Get it this way. It's supposed to be NH2. This one's got NH3 for some reason. Wow, I'm really screwing this up. There we go. Help if I colored by element, maybe. But right. Okay, so now that's our alpha carbon in the middle with a hydrogen kind of facing away from us. On the left is the amino group, although for some reason this is NH3 instead of NH2. But that blue sphere, that would be our nitrogen, so that's the amino side. On the right is the carboxylic acid. It's COOH. So one of these oxygens should actually be double bonded to the carbon, but unfortunately carbon doesn't, or Chimera doesn't show us double bonds. The other oxygen should really have a hydrogen on it, but it doesn't for whatever reason. And then this is alanine. It has just a methyl group as the R group. So this CH3 up at the top, the carbon and three hydrogens up at the top, that's the side chain for this particular molecule, or yeah, particular amino acid. Let me just pull up one other one. Here's phenylalanine. So let's see if I can get this oriented correctly the first time. That's better. Let me change that to ball and stick. Color by element. So once again, our alpha carbon is down here in the middle, and there's a hydrogen kind of hiding that's facing away from us off of that alpha carbon. The amino group is on the left. Once again, for some reason, it's showing NH3 instead of NH2. Over on the right would be the carboxylic acid group. So there's a double bonded oxygen and really should be an OH group over here. And then everything up at the top of this molecule is all side chain. So there's a methyl group here, CH3, but then one of the hydrogens has been replaced with an entire benzene ring. So if I go under atoms, bonds, and rings, and turn on aromatic circle, we get that little circle in here indicating the alternating double bonds that make this thing benzene. Right, so it's exactly the same as the alanine I showed you before, but with the addition of this entire benzene ring. One thing I didn't point out before, but probably, you probably noticed this, sometimes the side chains are bigger than the rest of the amino acid itself. All right, so those are just a couple of amino acids. Let me show you a couple of secondary structures. So this is actually keratin, which consists of nothing but an alpha helix, just really, really long alpha helices. And so right now I'm viewing it as a simple ball and stick model. Anybody notice anything weird about this model? Anything surprising about this? Think about valence. About how many bonds a carbon should have. What's missing? Somebody said it. It's the hydrogens, right? This doesn't have any hydrogens. They're all missing. And sometimes that happens with these models. It's kind of like that structural formula model, or that shorthand notation for illustrating molecules where they kind of just leave all the hydrogens off. So you'd have to be careful if you were using this model for rendering something or using this as a reference. You'd have to be aware that, oh my gosh, something's missing here. And there is a tool in Chimera that allows you to put those missing hydrogens on. And I don't remember if I showed you this before, but under the tools menu in Chimera, there's a whole lot of things that let you modify molecules. And one of them is called structure editing. And the very first one is add H. This brings up this little separate window. And usually I find that the default settings work fine. If I click OK, it should hopefully do that. Adds all of the missing hydrogens. Now, there's another way I could view this. Since I know that this keratin model is an alpha helix, I could choose to just view the alpha helix, and I could do that by turning on ribbon representation. So if I go under Actions, go to Ribbon and Show, this will turn on representation of the secondary structure. So there's a ribbon representation, and then I can hide all of the other atoms. So that little corkscrew right there, that is the structure of the alpha helix. And then I also have a beta sheet. So these are some beta sheets. And notice that they automatically come in a ribbon representation. And there are arrowheads on the strands of the amino acid chain that show the direction of the amino acid chain. They're always pointing from the N terminus towards the C terminus. This is actually the structure of silk. Silk is nothing but a bunch of flat beta sheets. But just to show you that you could actually view these as a bunch of separate atoms and bonds, if I say show atoms and bonds, we see a bunch of atoms. I can hide the ribbons. And you see that these really are a whole bunch of amino acids and molecules. Let's see if I change that to ball and stick and color by element. A little hard to tell, but there are distinct sheets of that amino acid chain in here. And we're also missing our hydrogens again. So I would want to go onto structure editing, add H. That adds a whole bunch more atoms in there. It gets a little confusing at this point. So you can see why the ribbon representation is so helpful if what you're interested in is trying to simply follow the amino acid chain as it winds through the molecule. So all of the atoms that are sticking out from these ribbons, those are all the side chains that stick out to the side of the amino acid chain. So we just looked at keratin. I was going to show you some fibrous proteins. We looked at keratin, we looked at silk, which actually are considered fibrous proteins. Here's a different fibrous protein. This is collagen. So collagen consists of just 3 sets of alpha helices that are intertwined, that are braided. So if I turn on the atoms and bonds and show them, what you're seeing here are the side chains sticking out. If I hide the ribbons, you see this is the structure of collagen as represented in this case by a ball and stick model. A little harder to tell that it's 3 braided strands here. Well, I wonder. There's a handy selection menu in Chimera. And one thing I can do in Chimera is I can say select by chain. Okay, so conveniently, the 3 strands of the collagen molecule are actually designated separately in here. So I can select the individual chains. So I could say select chain A, give it one color, select chain B, give it a different color, select chain C, give it yet another color, and then select clear selection. So then you get a better sense that this really is 3 intertwined chains of the amino acid chain. And if I switch this back to the ribbon representation and hide the atoms, it keeps that coloring by chain, which is kind of handy. A different structural molecule, myosin, this would be the molecule that makes up what we call the thick filament in muscle. So this is actually more of a compact globular protein, but what you're looking at here is the myosin head. So the skinnier part of the molecule to the left is part of the actual filament, the thick filament, and then this big blob over here on the right, that's the head of the myosin molecule that actually attaches to the actin to create that power stroke for contraction of muscles. And I think there's a whole bunch of chains here. If I go to select chain, well, there's at least A, Y, and Z. There must be water molecules in here somewhere. If I say select the water, yeah, there's like a bunch of tiny little red dots, which are some water molecules that are part of the model. Under actions, atoms, and bonds, I can actually delete those if I want to get rid of them. But then apparently there's some other things in here that aren't really part of the myosin. There's something in here that's referred to as hetero compounds. Yeah, there's some little green balls. That's magnesium. I don't know why that's in there. And I don't know, that's supposed to be a carbon, but I don't know. If I wanted this thing to look more like the actual, myosin head that we're used to when we see, pictures of the filaments in a muscle cell, if I add a surface to this, now we get a sense for the overall 3-dimensional structure of the molecule. All right, here's an example of an enzyme. So this is a compact blob. This is the same one that I showed you in my PowerPoint. This is cyclooxygenase. So if I select, let me show you a really handy thing in Chimera. Is this selection menu is really, really powerful. It lets you select all kinds of different things. This molecule consists of 2 separate amino acid chains, A and B. So I could select just one chain, or I could select the other chain. I could do different things to the different chains if I wanted to. But I can also select things by chemistry. I can select individual elements. I can select functional groups like amines or aromatic rings or esters or things like that. I could even select individual amino acid residues. So if I wanted to select just the alanines that are present in this molecule, everything that's highlighted right now are just alanine amino acids. Or I could select a different residue like methionine. I don't know if there's any in here or not. Oh yeah, a bunch of methionines. So I can select, individual amino acids, but then under structure, I could simply select the entire protein. Now, you might think that's going to select the whole molecule, but you might notice that there are some rings in here. These are the heme groups, which are not really considered protein. So if I say select structure protein, that's selecting only the proteins and not selecting the heme groups. So now if I do something like add a surface, it's only going to apply that surface to the selected protein molecule. And notice my heme groups, they're maybe kind of hard to see up there. They're kind of small, but they're still represented as stick models. So then I could say select chemistry. Let's see, where was that? Is that under? Oh, if I go to select residue and I say select heme, heme is not, it's not an amino acid, but it's recognizing that it's a distinct structure. So I could select the heme groups and then make those sphere models and color by element. So then we can see the heme groups are sitting in these deep pockets on the surface of the protein. If I go back and select the protein, go select structure protein. If I want to color that, I could give a different color to the protein surface. And then those heme groups really stand out. So those are the active sites of this particular enzyme. This is a potassium channel protein. And coming in by default as a ribbon model, I could select just the protein. So again, select structure protein. So it's selecting only the protein and not anything else that might happen to be in here. I could add a surface to that. So there's my surface model of the protein. Now, when I rotate it and look at the bottom, notice you can look almost all the way through the thing. It's got a hollow pore that runs right through the very center of the molecule. Same thing if I look at it from the top view. Those purple balls, those actually represent potassium ions that are sitting in the pore waiting for their turn to pass through. I could colorize the protein, give it some pretty color. That. I know that I retrieved this model from a very interesting database that I'm going to show you on Wednesday. There's a whole database called the Orientation of Proteins in Membranes database. Database that allows you to download hundreds and hundreds of different membrane-bound proteins, like channels and receptors, and it'll actually show you how the protein sits in the membrane. I'll show you on Wednesday how you do that. I'm going to do it fairly quickly here. I need to select the right thing. Yeah, I need to select something called dumb and view them. And make sure I'm showing the atoms and viewing them as spheres. There they are. So notice the layer of red spheres and the layer of blue spheres that appeared now. The red spheres represent the outer edge of the lipid bilayer. So basically the outside of the cell, the outer part of the lipid bilayer membrane. The blue represents the inner part of the cell membrane. So by turning on these red and blue spheres, it allows me to tell exactly how this molecule is positioned within the cell membrane. The membrane itself would basically be right there where the blue and red balls are. I'll repeat this on Wednesday probably, but there's a really strange logic to knowing whether it's the red or blue balls that are outside or inside. You know, what's the inside of the cell? In this case, what's the outside of the cell? The way it works is that the word outside starts with an O, which is the symbol for oxygen. Oxygen is red in the CPK scheme. So the red layer is the O layer, that's the outside of the cell membrane. Inner has two N's in it, and it's for nitrogen. Nitrogen's always blue in the CPK color scheme. So the blue balls represent the inner surface of the cell membrane. So that's how you can keep straight what's inside and outside. So if I was doing an illustration of this protein, and I want to make sure I drew it right, so it's sitting in the exact right place in the cell membrane, I'd use this OPM database, this orientation of proteins and membranes database, because it always comes in with these layers of red and blue spheres. The other day when I first introduced Chimera, I said that there were some viewing controls. And if you click on any of these, it brings up this separate viewing window where we can control things like lighting, effects like shadows and silhouettes and depth cueing and so forth. Let me turn the shadows on. So now you see we got some cast shadows here. I can put the silhouettes on to get like little black outlines around things. There's something here I didn't show you the other day because it wouldn't have worked the other day. But there's something called side view. And what side view does is it gives you this representation of the molecule, and it shows you the point that we're looking at the molecule from. So that little yellow square on the far left basically represents the camera view, your eyeball. And the two yellow planes are planes that we can actually move to crop into this molecule. I'm going to try to, whoops, my thingy disappeared. I'm going to try to position this molecule a little better. It's tilting strangely. I'm looking at my side view window to try to get this thing so it's sitting perfectly straight up and down. Kind of like that. And then what I can do is I can actually move this left-hand yellow line and crop into the protein. And what that does is it allows me to create a cutaway view, basically cut off half the protein. So I'm now looking into the interior of the protein, and I can tilt it around a little bit here, which is going to change it slightly. Notice the little purple balls up at the top. We're actually seeing the potassium ions as they sit in the channel, but you can kind of tell now that there's an almost continuous channel running right through the middle of this molecule. And we're going to utilize this for your next larger exercise, I'm actually going to have you create a cross-sectional view of a protein like this, and to illustrate kind of the inner workings of the molecule. Let's see, what else do I want to show you in here? All right, well, here's a model of myoglobin. So this is a good example of showing the tertiary structure of a protein. Once again, if I say select structure protein, it's going to select all the protein, but it's not selecting the heme group that actually carries the oxygen in the molecule. I could add a surface to the protein. And so now you see the protein surface, and you see that heme group in there. And if I say select residue, heme shows up under my list of residues again. So that lets me select the heme group. Then I can change that to maybe a sphere model, color by element. So now we see how that heme group sits inside a single myoglobin molecule. Do the same thing with hemoglobin. And, oh shoot, all right, this is not quite the hemoglobin molecule that I wanted, because this is only half a hemoglobin molecule. There's only two chains here. Chain A, chain B. So hemoglobin is actually supposed to consist of 4 identical chains, or almost identical chains. There's a little difference in them, but this model, for whatever reason, is only giving me half of the full hemoglobin molecule. So I've shown you a little bit about what's under this select menu. So, I've shown you that you can select different chains if you have different amino acid chains. You could select things like individual elements or functional groups. I don't use that very often. You could select residues, like you could select, again, every glutamic acid in here. So there's a few that just got highlighted in green. Anytime you want to undo a selection or just turn it off, just hit clear selection. If I have a molecule that happens to have a heme group, it'll actually show up as a residue. But I can also select things like structure. So I could select, if I had any nucleic acid in here, this would allow me to select only the nucleic acids. If I have protein, which of course I do in this case, it lets me select the proteins. I could also do something like, I could say select and invert all models. So I just selected all the protein in the molecule. If I say invert, it's going to select everything that's not currently selected. So it's going to select everything that's not protein. So sometimes that's an easy way to select things like these heme groups, that's the only thing in here that's not a protein. So if you don't have the heme group showing up under your list of residues, you just select the whole protein and then select the inverse, and it'll select everything that's not protein. There are other, yeah, is there a question? No. Sorry. There are some other ways to select things in Chimera. If you hold down the control key on the Mac, and I'm trying to think what the equivalent of that is on Windows. But if you hold down the control key and then you click on anything in the model, it's going to select whatever part you're hovering over. So notice as I hover the mouse over part of this orange protein, it comes up with a little label. And I don't know, you may not be able to see that up there very well, but there's a little white label that pops up right where my cursor is. And it says SER 138.A. What does all that mean? SER tells me that this is a serine, which is one of the amino acids. So it's a serine residue. It tells me that it's at position number 138. So this is the 138th amino acid that was laid down in the amino acid sequence for this particular amino acid chain. And then the dot A is telling me that it's part of the A chain of the two chains in this molecule. If I hover over a different part, say on the blue one, this one says ALA 53.B. That tells me that that is an alanine amino acid at the number 53 position of the B chain of this particular molecule. And if I hover over part of the heme group, it tells me I'm hovering over heme number 1 in the A chain. Well, there is only one heme in the A chain. And it actually says it's C2B. It's carbon number 2 in the heme group. But if I hold down the control key and actually click on some part of this molecule, so I'm holding control and I click on that serine 138A. If you look closely, if I zoom in on this, maybe you can see it better, that it's highlighted this one little portion of the protein. It's highlighting the entire serine residue because I clicked on it with the control key. Then I can use the up arrows, or I can use arrows on the keyboard. There's an up arrow. If I hit that, it's now going to select whatever else that particular serine is part of. So that serine is part of this long amino acid chain. So if you hit the up arrow, actually, oh, I forgot about that. It actually does secondary structures. So that serine is part of this one alpha helix. So when I hit the up arrow, it selected the alpha helix that it's part of. If I hit the up arrow again, it should now select the entire amino acid chain that this is part of, which it did. But it didn't select the heme group. If I hit up one more time, it now gets the heme group. And I think if I hit up one more time, it's now going to get the entire molecule because now it's going to add the other chain, the blue chain. Let's see if it does that or not. Yep. So it just selected the whole thing. So the point being that you can click on any small piece of the molecule and then using the up arrow, just select progressively larger portions of that in sort of a hierarchy order. Let me deselect these and show you one situation where that might be useful. I wish I had the whole hemoglobin molecule here, but I'll make do just with this half a hemoglobin. You probably all heard of the condition sickle cell anemia. Sickle cell anemia is a single amino acid change in one chain of the hemoglobin. And it's a change from, it's like glutamic acid to alanine or glutamic acid to glycine, something like that. It's one amino acid changes. And the amino acid happens to be in the number 6 position of whichever chain it is. I think it's in the A chain. So let's say you wanted to illustrate sickle cell anemia and you wanted to highlight that difference. You'd have to search around the molecule and see if you could figure out, all right, where's the number 6 position on here? Well, number 6 tells me that it's got to be near one end of the molecule or the other. It's got to either be right near the very beginning or right near the very end. So I need to find a free end of the molecule. Well, if I hover over this part, it tells me that's a histidine at the number 143 position. So that's definitely not near the beginning. All right, here's another end down here. See this little end right there? If I hover over that, all right, that's valine at the number 1 position. If I move a little farther, there's a histidine at the number 2. There's a leucine at number 3. A threonine at number 4. A proline at number 5. Still proline. And there we go. There's a glutamic acid at the number 6 position of this chain of the protein. So let's say I wanted to highlight that. I could hold my control key down and click on it, and that's going to highlight just that one amino acid. And then, there's various ways that I could draw attention to this thing. I could simply change its color. Say, okay, that's the culprit right there. It's the change of that one amino acid. I could even change the representation of it. If I turn on atoms and bonds, it's going to do it only for what's currently selected. So this is showing me the side chain for that particular amino acid, that glutamic acid. I could make the atoms and bonds, a ball and stick or sphere. That kind of obscures things. So maybe that's not a good choice. Maybe going back to ball and stick is better. I could hide the ribbon so that we see only the atoms, although that seems like it's broken the chain, so maybe that's not a good choice. But anyway, and yeah, changing the color of that helps. So anyway, so that's going to draw attention to that one amino acid. All right, here's the culprit in sickle cell anemia. Let's check it. All right, hopefully this will open. Oh yeah, it did. This is the nucleosome model that I used to create a picture that was in the lecture earlier. So this is where 1 3/4 turns of the DNA double helix wrap around a cluster of 8 proteins to produce something called the nucleosome, which is the building block of chromatin. So I could go to my select menu, say select all protein. It's now selected only the histone proteins. I could add surfaces to those. Hopefully this will work. Sometimes Chimera has fits with these big models. All right, seemed to work. But the histones they're actually 8 separate proteins. So if I go select chain, I get, wow, A, B, C, D, all the way down to J. A is one of those proteins. I'm going to give that a certain color. Select chain B. That a different color. Select chain C. Give that a different color. So basically what I'm trying to do here is I'm trying to colorize all of the 8 histone proteins different colors. Where did I leave off? 1, 2, 3, 4. this different color. And I think one more. 1, 2, 3, 4, 5, 6, 7, 8. One more color. All right. So my 8 histone proteins are all now colored different colors, but I've still got my 1 3/4 strands of the DNA double helix wrapping around here. I could select those. Those are the 2 remaining chains here. So those are chains I and J. Those are actually the chains of DNA. Or I could simply say select structure nucleic acid. And that automatically selects all of the DNA. And I could now view those as ball and stick models, or I could view them as sphere models. I think they're missing a bunch of hydrogens. Let me try my structure editing to add the missing hydrogens. See if that does anything. Oop. I may have made it mad. Well, that's fine. Whoops. Still thinking. I could color the DNA by element. Anyway, the point being that I can try to distinguish the DNA from the proteins. There is actually another fun thing under the tools. Under tools, what's it under? Depiction, I think. Yeah. Under tools, depiction, nucleotides, there's actually a whole menu to bring up different ways of viewing DNA or RNA, any kind of nucleic acid. So I could show the backbone as a ribbon. I can show everything as like a ladder. If I apply that, let's see what happens here. There we go. So that's a way of creating this sort of ladder representation of DNA if I wanted to. So anyway, just a way of showing the histones differently from the DNA. All right, one last thing I want to show you here in Chimera before we wrap up for today. One really handy feature of Chimera is that it lets you retrieve models directly from many of the online databases without even having to open the database. Now, this depends on one important thing. You have to know the code number for the model that you want. So on Wednesday, I'm going to show you how to search things like the protein data bank to find the model that you want. But assuming that you somehow already know the code number for the protein you want, you can retrieve it directly from the protein data bank or from a number of other databases. So if you go to the file menu, instead of saying open, or instead of simply double clicking one of the files that I have on my desktop, we're going to go File, Fetch by ID. And that brings up this window. And all of these things listed on the left are different databases of molecular model files. And I'm going to show you a bunch of these on Wednesday. So up at the top here, the NDB, this is the nucleic acid database. So this whole database of structures of DNA, RNA, messenger RNA, transfer RNA, etc. But you have to know the right ID number to put in there. This one right here, PDB, that's the protein data bank. And it just so happens that I know a handful of these code numbers off the top of my head. because I've used them so many times. I'll come back to that in a minute. I'll actually enter one. PDB MMCIF, it's the same thing. It's accessing the same molecules from the protein data bank. It's just pulling them in a different file format. PDB BioUnit is also the protein data bank. And I'll show you on Wednesday that when you download a model from the protein data bank, you typically have two choices. You can download the basic model, or you can download what's called the biological assembly. So hemoglobin's a good example. If you download just the basic hemoglobin molecule from the protein data bank, oftentimes it comes in with only 2 out of the 4 chains, like the one that I opened up earlier. If you say download the biological assembly, it's going to give you the functional molecule. So it's going to actually have to have all 4 of the amino acid chains. I forget what some of these are. UniProt, that's the Universal Protein Database. ViperDB is fun. I'll show you that on Wednesday. That's the Virus Particle Explorer. It lets you download what's called the nucleocapsid of lots of viruses. So you can actually get like a complete model of what's called the nucleocapsid of a virus. PubChem I'll show you on Wednesday. That's one for smaller molecules. EMDB is the European Molecular Database. So let's go back to just plain old PDB, the Protein Data Bank. And there's one that I've used probably 100 times. Used it a lot in my own work. This used to be a common one that I used for the chemotherapy work that I used to do. One tub. One tub is tubulin. Tubulin is the protein that makes up microtubules in your cells. Microtubules are part of the cytoskeleton that helps give cells their shape. It's also the thing that forms the mitotic spindle. And tubulin is really, really important in chemotherapy because there are several different chemotherapy agents that specifically target the microtubules to stop cells from dividing, which is one way of fighting cancer. So if I simply enter that 4-digit code, 1TUB, no, there has to be. I've used it 100 times. Try that again. Why are you not working? PDB. Really? All right, either the protein data bank is having issues. There's no way that that's the wrong one. I'm actually going to go in there just to check. Yeah, there it is. 1TUB. What are you telling me that it doesn't exist? I've never had to do this before, but let's try all caps. All right, you're just being glitchy on me here. Maybe if I quit Chimera and try again. I mean, I'll just try a different one if this continues to not work. All right, that's weird. Let's try 2HBS. I think that's right. All right. Don't ask me why this refuses to work. Something glitchy with Chimera here. I mean, here's tubulin that I was trying to download. I'll come back and show you this in more detail on Wednesday, but there's a little thumbnail picture of tubulin. Tubulin is a dimer, meaning it's a 2-part protein. There are 2 different amino acid chains. And there's what's called alpha tubulin and beta tubulin. And the 2 of them have to work together in order to be a functional molecule. From the protein data bank, there's a button to download files. And I can download it in PDB format, or I can download the biological assembly, which is the fully functional molecule. I think for this particular one, it doesn't matter. Let me try the biological assembly. And I open that. Now let's try opening that in Chimera. Oh, I don't know why it opened Chimera X. Let me fix that. Open with regular Chimera. It's thinking. Maybe. Chimera is not playing nice today. For some reason, it does not want to open this molecule. All right, finally opened it. So this is tubulin. And so it's a two-part molecule. It has two very similar chains. This is the beta chain on the left, and this is the alpha chain on the right. And I could select those individually, just like I did before. I could color that, put a surface on it. All right, didn't like that, but it seemed to work. I could select the other chain, give that a different color, give that a surface. Yeah, it worked. All right. I can make these surfaces transparent. It shows you can still see the ribbons inside. I'm going to select the proteins. I'm going to make sure that any atoms that are part of the proteins are hidden. So for some reason, there were some little side chains showing. But then I'm going to select, there are some other molecules in here. Again, these are not amino acid residues, but they still show up in the list of residues. There's something called GDP, which is guanosine diphosphate. So it's like ADP. So that's over here on the left, right there. Let's see what else we got. We got, there's also a GTP, so guanosine triphosphate. So that's like ATP, but based on guanine instead. And then there's one more molecule in here, something called TXL. That's Taxol, or sometimes called Taxotere. That's a very potent chemotherapy drug. So this is a chemotherapy drug that acts by binding to the tubulin protein in the microtubules. And actually what it does is it prevents the mitotic spindle from breaking down at the end of mitosis. So the cell replicates its DNA, it goes through mitosis, the chromosomes migrate to opposite ends of the cell, and then it stops. And it can never actually complete the process of cell division because the microtubules won't break down. And so eventually the cell just dies. So the cell dies stuck in mitosis. So it's a good way of targeting any cells that are rapidly dividing, rapidly multiplying, which is great if you want to kill cancer, but it's also why your hair falls out when you're on chemotherapy, because it also targets the rapidly dividing cells in the hair follicles. It's also one reason why people get nausea when they're on certain chemotherapy drugs, because it also targets the cells that in the digestive tract, which are also undergoing rapid cell division. So the bad side effects of many chemotherapy drugs are related to the fact that many of them, what they're doing is they're targeting any cells that are undergoing rapid cell division. Right, so you can see Chimera is very powerful, in letting you select all sorts of different things, change the colors of things once you've selected them, change the style, ribbons and atoms and bonds and surfaces and all sorts of things. And then if you want to save any images out of Chimera, we looked at this briefly the other day when we were doing the Illustrator thing, but you just go to File, Save Image. By default, it comes up just exporting a certain number of pixels. So in this case, it's going to give me an image that's 3,184 pixels wide, 1,864 pixels tall. I'm just more comfortable thinking in terms of, inches and things like that. So I'm going to click Use Print Units. And so now I can specify, I want a certain width, like 12 inches. As soon as I hit the Tab key and move into the next section, it's going to automatically adjust the height because I have a box checked here, maintain aspect ratio. So as I reduce the width, it reduces the height proportionally. And then I can set my resolution here in however many, pixels per inch that I want. And then just click Save. We should get an image. Here's my ping image. Chimera will actually produce publication quality images. I mean, if you just want, with our Illustrator molecule, we wanted a vector image, which is why I had to redraw it in Illustrator. But if you just wanted a raster image, you can export publication quality raster image, pixel-based images directly out of Chimera.